What's up, AP Bio students? Today, I'm going to drop some knowledge on you about genetics. I'm in Johnstown, Pennsylvania, hitting up some of the hottest biologists, and we're going to teach you about linked genes and genetic mapping. Let's go! Before we get into our lesson, I'll give you guys a brief overview. There's millions of genes located in the human DNA, and a lot of those genes get inherited together because they're located next to each other on the chromosomes. These are known as linked genes. Now, a lot of you may say, wait a second, that goes against Mendel's law of independent assortment. What? I'm here with geneticists Alex and Hayley, and they're going to break down to us how linked genes affect inheritance. They're even going to provide us with a fire example. Take it away, guys.
Okay, this is an example of a homologous pair, and we only portrayed a few of the genes on here, but particularly down here, this is an example of linked genes because of how close they are.
Alright, so a guy by the name of Morgan designed an experiment with wild type of flies that normally have gray bodies with normal wings. Alright, we're going to represent gray bodies with little b plus, and normal wings with VG plus. And through breeding, he obtained mutant flies with black bodies and short wings, both recessive to the normal genes. The black bodies are represented with a little b, and the short wings are represented with a VG.
So, for the parental generation, Morgan crossed a gray body normal winged fly with a black body short winged fly. So that was the P generation would be B plus, B plus, VG plus, VG plus. And that's the gray body normal wing crossed with little b, VG, VG, and that's the black-bodied short wing. Then for the F1 generation, the results were a dihybrid of gray-bodied normal wings, so it was B plus, little b, VG plus, VG.
And from that, he crossed another mutant black body short wing fly. And that'd be little b, VG, VG. And with this one was a male and this one was female. That's because the male always contained the recessive short wing and black body genes. So from that, we got our test cross results. Alright, so here are the test cross results. Here we have the sperm, which is always the BVG since it's recessive, but the eggs could be any of these. They could be both dominants, both recessives, dominant and recessive, or dominant and recessive. And these would be the results that you would get. The predicted values here show us that if you got a 1 to 1 to 0 to 0, it would be on the same chromosome, theoretically. And if you got a 1 to 1 to 1 to 1 for all these results, it would be on separate chromosomes. And the actual results he got were B plus VG plus 965, BVG 944, and B plus VG 206, BVG plus 185. This shows us that these two, as you can tell, are significantly higher. This is because of because they're on the same chromosome, as you can see here, even though they're not exact. That's just what it shows. Alright, so the phenotypes of the parental, as you can tell, were these two right here. This shows us that you get normal wings, gray bodies, and short wings, black bodies. And these were the highest number of the offspring that we received. But here you get gray bodies, short wings, and here you get black bodies, long wings. and those are not from the parental generation. So they were not, they did not show the numbers that the parental generation showed. So this gives us the conclusion that the predicted values gave us are that they are on the same chromosome.
These non-parental phenotypes show us that the genes are not actually linked together, and this brings the notion of genetic recombination into play. Since the majority of offspring had the parental generation's genes here, Morgan concluded that body color and wing size are genetically linked on the same chromosome. Non-parental phenotypes indicate a mechanism that occasionally breaks links between the genes.
I'll tell you guys, I was dying about Next up, we're going to hit up genetic recombination. I'm here with Josh Hilands and Karsen Bailey, and right now, we're going to go over genetic recombination.
Genetic recombination is the production of offspring with a combination of traits different from parents. Parental type is offspring with the same combination of traits from their parents. Recombinant types are offspring with different combinations of traits from their own parents. Recombinant types are exhibited in both linked and unlinked genes. Recombinant types are much more common in unlinked genes. Recombinant chromosomes result in increased genetic variation, which helps natural selection.
I tell you, man, that was the bomb.
Okay guys, so here's an example of recombinance with unlinked genes. We're going to cross two pea plants. One will be dihybrid and one will be homozygous recessive. And the dihybrid one will form 4 types of gametes. You can see them here. And the homozygous one will form only 1 type of gamete. And they will produce, after fertilization, they'll produce 4 different types of offspring. And 2 of them will have combinations of traits that are the same as the parents, like for example round and yellow. One of the parents was round and yellow, this parent. And then for wrinkled and green, one of the parents actually was wrinkled and green. But then the other 2 types of offspring will be recombinant types, which will mean that the combination of traits will be different from the parents. Like wrinkled and yellow right here, neither of the parents were actually wrinkled and yellow, but it can still happen. And parental types and recombinant types are both 50% likely. So now I'm going to go over an example with linked genes. For this cross, the recessive traits are black body and vestigial wings, and the dominant traits are gray body and normal wings. And we're making fruit flies here, by the way. So the female in this cross has a gray body and normal wings, and the male in this cross has a black body and vestigial wings, which means can't use them. So for the female, it starts out with these 2 chromosomes here, and then they replicate and cross over, and the chromosomes actually end up with different combinations of these alleles, and that produces 4 different types of eggs. 2 of the eggs would be parental type eggs, like parental type chromosomes, I mean, and then 2 of the eggs would have recombinant type chromosomes. And then for the male, since it's all the same alleles, the crossing over wouldn't produce any different type of combination, so he just produces 1 type of sperm. And then when fertilization happens, you'd get 4 different types of offspring, and then as you can see by the numbers, the parental type would be a lot more common than the recombinant type.
Last but certainly not least, I'm with my main man Drew, and he's going to fill you in on what you didn't know about genetic mapping.
I'm here to tell you about genetic mapping, and we can credit Alfred H. Sturtevant for a way to map genes on chromosomes, which is his idea of genetic mapping, which is an ordered list of genetic loci along a particular chromosome. He created this idea of genetic mapping based on his hypothesis that recombination frequency is dependent on the distance between certain genes on a chromosome. He believed that the farther apart these 2 genes are located on this chromosome, there's a higher chance of the crossing over to occur in the space between these genes, which makes sense because if you have a larger space in between, there's a higher possibility at each location in that space that a crossing over will occur. One such uses of this is in a linkage map, which maps out how the chromosomes are located, how the chromosomes are built, based off the recombination frequencies that each gene has. And these linkage maps utilize a type of data that was created by Sturtevant called a map unit, which is basically just a 1% recombination frequency for each one. So it's basically split into 100. In some situations, though, you'll have two genes that we can use linkage maps to discover this. That two genes can be located so far apart on a chromosome that even though they are actually located on the same chromosome, the possibility of crossing over is so high that basically they're on opposite chromosomes. So even though they're located on the same chromosome, you can almost basically identify them as being located on separate ones. There's another example of linkage maps that can be used, which is a cytogenetic map, which instead of using recombination frequencies to locate them on the map, they'll correspond certain genes in certain loci to certain giveaways on a chromosome, such as if there's a stained band on the chromosome, they'll just locate this gene next to the stained band. It's another way to just basically map out a chromosome utilizing genes.
Thanks for watching. That's all we got. Join in next week when we talk about chromosomal disorders.